Success is never owned, it is rented, and the rent is due every day. At Agappe, every day we plan to do something new, something that helps us to succeed, to be the best partner in the diagnostic field for the welfare of mankind. This enables us to fulfill our promise on quality and innovation, thus providing the best solution to our customers. Just as a gem cannot be polished without friction, we keep developing our skills by setting our standards higher and higher. Today, we are here to witness the launch of molecular diagnostic solution amidst COVID-19, which is comparatively faster and much affordable one. I welcome you all to this grand occasion.
Hi all, welcome to the grand launch of Molecular Diagnostics Division and nCoV detection's new advanced testing. Reducing the turnaround time of the confirmatory molecular testing for COVID was a dream 2 to 3 months before, which is now a reality. We believe that RT-LAMP technology can trigger a revolution, bringing affordability into molecular testing. What we do at Agappe will make an amazing impact in today's COVID testing, making every laboratory capable of having a molecular diagnostic setup, thereby effectively handling similar situations in future. With this goal in mind, we wanted to take our R&D to the next level to compete with the multinational brands.
At Agappe, we find innovative solutions using molecular technologies to diagnose infectious diseases. To fight against this pandemic, we did our best to have a real-time RT-LAMP based COVID testing technology, which is cost-effective, fast, simple, yet highly accurate, and a real-time RT-LAMP analyzer to detect that assay. Taking up such a project and working as a part of India's largest reagent manufacturing facility is the most exciting part of it, which sets the tone for the products under development and to have a sense of quality and precision.
When we thought about making the COVID testing more innovative, and affordable solution is what we had in mind. Today, Agappe is ready to trigger a revolution in diagnosing the novel coronavirus disease. It has been a long time since something like that happened for molecular diagnostic segment as a whole in India.
We are sure that RT-LAMP technology based assay and detection instrument will reduce the turnaround time and provide high sensitivity, specificity and accuracy equivalent to currently used technologies. The very important step in our strategy is to prove that RT-LAMP instrument is better than any other portable or point-of-care methods available in India. Meeting all expectations of a common man on COVID testing, our technologies will score more. With the ambiguity prevalent in COVID detection today, there is a huge demand for RT-LAMP, that is reverse transcriptase loop-mediated isothermal amplification technology.
The new highly compact RT-LAMP reaction-based equipment and real-time detection kits for COVID testing is launching today. I thank all doctors, well-wishers and those who have been involved directly or indirectly in the product development for all your hard work. I am really excited to reveal RT-LAMP based Mispa Lume and the real-time COVID detection kits LumeSCREEN-nCoV.
are really enthusiastic to learn more and explore about molecular diagnostics. New ideas really came faster. However, briefly, we wanted to create, communicate, and deliver something that's perfect. With Mispa Lume, we stand a great chance of avoiding major investment for setting up a molecular laboratory.
Currently available molecular diagnostic platforms have a huge infrastructure input and are highly priced. A compact and affordable solution is the need of the day.
RT-LAMP is a one-step nucleic acid amplification method to multiply specific sequences of target RNA. It combines reverse transcription, amplification, and detection. This is performed at a constant temperature of 62.5 degrees Celsius for 35 minutes. With the isothermal reaction in Mispa Lume, using LumeSCREEN-nCoV provides many advantages over the real-time PCR technology. Technically, Mispa Lume is an isothermal amplification analyzer that has an accurate heating module which maintains the required temperature and an optical detection module to detect the amplicons on real time and plot it in an amplification plot, so that our customer can monitor the amplification of the sample in real-time condition. So expensive real-time PCR machine is not at all required. Delays in testing due to long procedure also make it difficult for public health officials to tailor their responses to the right people, those who are infectious and people to whom they may have passed the virus. So with Mispa Lume, we provide the shortest time to result.
There is a need for reliable, economical, rapid tests for COVID diagnosis. The present-day techniques takes longer time for diagnosis and also report false positive and false negative results. In this context, your contribution is highly significant. I'm glad to know that you have developed a rapid diagnostic kit using new technology, that is RT-LAMP, which is 10 times sensitive compared to the present-day RT-PCR technique. I am also glad to know that ICMR has given a nod for this procedure. Your contribution is highly commendable, and it goes a long way in the years to come in diagnosis. If economical, this test can be used for population screening and high-risk groups in particular for COVID diagnosis. I wish you all the best in your future endeavors. Best of luck. God bless you.
The assay is too fast. Positive results can be obtained in 10 to 35 minutes. COVID-19 testing centers can release report in 35 to 45 minutes after RNA extraction. So, sample to report turnaround time will be around 1 hour with very high accuracy of 99.35%.
Thanks to the latest isothermal loop-mediated amplification technique, abbreviated as LAMP technique, any of the labs can adopt this technique for COVID testing. There is no requirement for a costly infrastructure or machineries. Any technical staff in the lab can conduct the test taking the safety precautions. As the test seems to be highly sensitive and specific, the quality of the test report is as par with RT-PCR. The test may even replace antigen test, which has got a lot of limitations.
Yes, quality. At Agappe, there is no shortcut to quality. Diagnostic tests have optimal value only when the community is fully engaged and comply to participate within confined measures like it happens now. A global solidarity towards COVID test access to optimally combat current and future pandemics. The new product segment of LumeScreen guides the medical fraternity in the choice of therapy and follow-up of therapy success. Primers are designed based on CDC and WHO guidelines by targeting N gene and RDRP gene respectively. LumeSCREEN-nCoV has a limit of detection up to 4 copies per microliter with a sensitivity of 98.7% and 100% specificity. Positive prediction value is 100% and negative prediction value is 98.8%. Our RT lamp assay is listed in ICMR website under real-time RT-PCR technology.
As we already know that COVID assays which are already developed on the platform where you need to transport the material in dry ice and then take all the reagents in a right condition before they arrive to the site where they are going to be tested. The biggest advantage of having a COVID lamp test is that it can be performed and transported at the ambient temperature, and then it's going to be carried out on a simple machine which is can be a butter bath also. So for the conditions which we have in India, it's very much required to have a test which doesn't require any cold chain management, and it is matching to the requirement of our rural areas and then the primary healthcare centers as well. So the advantage of COVID lamp is that it is a very specific test which is going to be equivalent to the RT-PCR and which can be transported at the room temperature without any shift or changes in the outcome of the result.
The ready-to-use single reaction vials with lyophilized master mix can be stored at 2 to 8 degrees Celsius. Just add primer mix and RNA sample to complete the assay setup. No master mix preparation or aliquoting as in conventional methods, which need skilled technicians and prone to human errors.
The major problem with these reagents is the transport. Transport and maintenance of temperature. Minus 20 degrees centigrade temperature maintenance is a big challenge. So any reagent which we are procuring for PCR is actually from overseas, and so the entire transport process is very cumbersome. So you need something which has been initiated in India, has been processed in India. That is, in major cities like Kolkata, Mumbai, Delhi, it will not be a problem because it will be delivered easily. But what about the rural areas? The rural areas are very deep interiors. The transport time is so high and maintenance of this cold chain is very difficult. So anything which is prepared inside India will take a less transport time, and therefore the reagent stability and all these things will be much more assured, I suppose. So we definitely need some PCR reagents of PCR manufacturer in India who are manufacturing, and that will be beneficial for us in every sense.
LumeSCREEN-nCoV has lyophilized master mix in the cap of the reaction vial. So, customer has to add only the primers and RNA into the reaction vial, mix and load in Mispa Lume. Unlike real-time PCR reagent storage in minus 20 degrees Celsius, LumeSCREEN-nCoV kit can be stored in 2 to 8 degrees Celsius itself. The reagents are stabilized in a way that they can be transported at 2 to less than 30 degrees Celsius. LumeSCREEN-nCoV is compatible with any commercial RNA isolation kit. Summarizing it all, yes, we are ready with a highly specific and sensitive confirmatory real-time COVID test kit with simple process and user-friendly assay protocol using lyophilized reagents that can be stored at 2 to 8 degrees Celsius. compact equipment and simple assay workflow enables to handle samples even in remote areas.
At Agappe, we keep things simple. Thus, a confirmatory, accurate and user-friendly molecular diagnostic solution is in place to address the COVID-19 pandemic. The RT-LAMP technology is an innovative gene amplification technique which is ideal for Indian conditions. We are confident to build the national testing capacity above the currently available standard with the new LumeSCREEN-nCoV COVID testing kit. Let me express our deepest gratitude to all the doctors and medical practitioners who relentlessly supported and guided us for this noble cause. We strongly believe that the combination of Mispa Lume and LumeSCREEN-nCoV will make COVID testing truly revolutionary. We wish and pray that our humble contribution to the nation, especially to the diagnostic industry, will help you to serve the humanity better and faster. Thank you.